in a sentence, we kill cells based on what they're thinking. So more specifically, their transcriptional activity. And I think before I go too far into this, it's worth kind of staking out the broader context in which we're doing this. And people have endeavored to ablate solid tumors for a very long time, initially with things that were quite inspecific, like knives or radiation that kill effectively anything they touch, and then later, various small molecule poisons, and now, more recently, what we call targeted medicines, things like monoclonal antibodies and CAR T cells, of course. But I think it's worth pondering for a second that these are not really as targeted as they seem. Like I say, they're basically the biological equivalent of killing every redhead in the room, and which seems a little absurd, and it would be in any other context, right? You're effectively killing a cell based on its skin. And so we thought that there's a better way to do this, and basically to kill them based on what they're thinking, and it's predicated on this idea that a cancer cell knows it's cancerous. And consider for a moment that a lot has to go wrong by the time you have cancer. I mean, you have DNA repair mechanisms, you have tumor suppressors, you have immune surveillance, all those had to fail. And I'll come back to this in a moment, but this is based on the idea that the cell has its own detection for damage, and we can exploit that. But I guess if you're going to take one thing away from this slide, it's that we've taken fundamentally about the exact opposite approach to traditional drug development. We're effectively targeting things with information instead of chemistry. And so how do we do this? The base case is pretty simple. We basically put a suicide gene, in this case caspase-9, under a promoter that we want to kill. In a cancer, this is a truncated version of p53 for the most part. We've built a few versions of this targeting different things and also using multiple inducible caspases and self-activating caspases. But the idea is that you deliver this to the cell and it dies. And what's really cool about this technology is that you can implement Boolean logic in DNA. So if I wanted to kill, hypothetically, every p53 myocyte, but not a cardiomyocyte, that's pretty easy to do with a genetic program. It's very difficult to do with chemistry. And in the case of p53 in particular, I think something really interesting occurs in that, basically, if p53 is working, it's hard to have cancer. It's the guardian of the genome, right? It arrests cell division. And so most cancers mutate p53. And something really interesting happens, though, that when they mutate it, the cell frequently tries to rescue it by elevating levels of p53 transcription factors. This is kind of this concept of it knowing something's wrong. Like it wants p53, can't get it. And so even if you ablate p53, the transcription factors for it tend to go up. And so this is just a little in vitro data. I'll go through it quickly, but it kind of illustrates the point. This is a colon cancer line, and the version of p53 ablated has upregulated expression of our construct in this case. And this is one that's expressing luciferase, just to measure the expression going up. And kind of a quick example that if you use this to transfect both healthy and cancerous cells, the healthy cells are left unharmed, and you can not only see the expression come up in the cancer cells, but you can also kill them with the inducible suicide gene. Okay. That's not very good. It's not advancing. Can you guys move it forward? It's too early in the morning for this kind of problem. All right, there we go. Okay, so this is a cool bit of eye candy, and hopefully it will behave. It's time-lapse microscopy going after a colorectal cancer line. This one's P16 positive, so it just kind of illustrates we can target multiple things, but you can do it with any promoter. The one on the left, which in theory if it starts, so shows the cells dying really nicely. You can note the little apoptotic bodies forming on it. So in this case, they were transduced with a vector, and then we activate the suicide gene. What's really cool, and probably more important though, is that in the context of P16 negative cells on the right there, only the positive cells are harmed. So we're not seeing apoptosis in any of the P16 negative cells in this case. Okay, so at this point, you should have a burning question of how does one use this in vivo? And so we deliver this with a really exotic kind of lipid nanoparticle. And I'll go into some details of it in a minute, but the takeaway is that it's easy to make, it's not immunogenic, so we can do it multiple times, and it's well tolerated at really massive doses. So far, 10 times the human equivalent dose in non-human primates. And it's also relatively simple to make it in GMP at really large scales on a really cool microfluidic system. So for those of you who aren't familiar with the lipid nanoparticle technology in general, most of them gain entry into their target cells based on charge chemistry. They're cationic or conditionally so. And this works, but it's also quite toxic. So as the concentration goes up, it disrupts the membranes badly enough that the cells die. And this is particularly harmful in vivo, where they concentrate in places like the liver. Ours, on the other hand, is neutrally charged, so it doesn't have any of that toxicity, and instead it gains entry with a really interesting fusogenic peptide. And I don't have time to go into the details of this at this talk, but it was isolated from a reptilian orthoreovirus and modified heavily over the last decade or so. But it's the shortest or smallest documented fusogenic peptide that's sufficient for membrane fusion by 2 orders of magnitude. So it's super tiny. And this is really why it's not immunogenic. It sits there hanging out in the membrane of the nanoparticle. And when it comes in contact with the cell, it basically mixes the lipid of the nanoparticle with the cell. So it bypasses the entire endocytotic pathway. There's no receptor-mediated endocytosis, no targeting. It's utterly indiscriminate. And this kind of goes to this general theme that we're trying to target with the information, not the chemistry. So this is just illustrative of the issue with cationic lipids. You can see at low doses, they go into cells fine and they work well. As the dose increases, all the cells die, and that's not very helpful. So people have been trying to use lipid nanoparticles for quite some time. If you've been following that, the early versions were wildly toxic. Most of these things failed at phase 1 or 2. And even the newer ones that are like conditionally charged are really struggling to go beyond, say, the liver or like a DNA vaccine type of context. And but if you use neutral lipids, you can, I don't think DLT has actually ever really been reached in a human on it, but we've pushed it up over 10 mg per kg without any trouble on it. So these are massive doses of DNA. We're in the highest one we pushed was going up to about 1 gram of DNA in organisms. It's really high. So okay, on to the fun stuff. We started this, the first oncology stuff in prostate cancer, because our chief scientist, John Lewis, runs a prostate cancer research group and University of Alberta, Edmonton. And so we did a prostate cancer run first. And initially, we'd injected this intratumorally, which is kind of a compromise between some key opinion leaders and myself who thought it should be IV right away. But the results were really astounding. 90% reduction in tumor burden almost right away. You saw it in time lapse, most of the cells die in a couple hours. But in this case, we measured it 48 hours later. And it was striking enough that even our own scientists were very reluctant to talk about this until they'd repeated it a lot. And fortunately, it was quite reproducible. So these are now all IV administration, or almost all of them. And it's quite consistent. Like in this case, they're receiving the nanoparticle multiple times, but we don't do that anymore. And you can see the drop is where the dimerizer is added to activate the suicide gene. And even in an immune deficient model, a single treatment can basically double the lifespan of the animal. So it's pretty impressive. And of course, you can repeat dose. So this is just another piece of data, and I think it's actually pretty helpful. So this is targeting lung cancer at H1299, and so it's p53 positive. And what's cool about this is we're going to show you that it's very, very selective. So it's killing the tumor, and that's great. But this is some IVUS showing both that the tumor is lit up over there. I don't know if I have a laser. Oh, I do have a laser pointer. And what's really cool is that there's almost nothing like you can see targeted that isn't p53 positive. So you're shrinking the tumor, you're killing it. This is IV injection of a pretty good dose. And you're not getting any discernible off-target effects, but you're getting substantial ablation of the tumor. This is just one more kind of illustration showing tunnel staining. So this was an animal that was treated, and the tumor was taken out in section and looked for apoptosis. And you can see what just is interesting about it is that the red cells there are diffused through the tumor. So we're getting good penetration with this through IV administration. And we did this in metastatic tumors. It works great. In fact, even with a really aggressive metastatic cancer like B16 murine melanoma, a treatment on this for a week reduced metastases by 20-fold. So it's actually really effective at getting the metastases because they're basically defenseless. And this is it's an exciting piece of data, but it's really early. And but this I think shows kind of the the potential of this and where we're likely to go in the future. And so in this case, instead of just making a suicide gene that killed the tumor, we also express CD40 ligand. And we've done this with things like GM-CSF, tetanus antigen, influenza, and you can effectively turn a tumor into a vaccine against the tumor. And the idea here is that you could make, an immunologically cold tumor hot. And so we are doing studies now where we're pairing these with things like checkpoint inhibitors and seeing kind of better trying to characterize the tumor microenvironment. But I think it's really unique in that you can deliver this thing systemically and not only kill a target cell, but you can alter its behavior in ways that could elicit a more durable immune response. The biodistribution of it is awesome. This is IVUS data in mice with just a RNA. And then this is a PCR-based biodistribution in non-human primates. And what's really fascinating about this is, so the DNA we administer is a plasmid. It's non-integrating, it's transient. But it's also cleared very quickly, faster than I expected, to be honest. Because I expected it would be silenced relatively fast, but persist a bit longer. But you're looking at, what's that, like 8 logs or 7 logs of reduction of this vector over the period of basically a couple weeks. And so it doesn't accumulate in ways that are meaningful, and since it's just carrying a suicide gene anyway, if you activated it later, it would result in a dead cell, not a neoplasm. For the team, I guess I'll highlight John Lewis since he's here. I already mentioned his group also, Entos, had developed the lipid nanoparticle technology that we use for this. And so we started out initially going after actually senescent cells and going after oncology as first clinical target because it makes sense from a regulatory point of view. And so I've got a great team, experienced biotech people. And I think I'll wrap up there or leave a minute or so for questions. But I guess the takeaway is this is about as far different from the standard approach as you can get. I mean, we're effectively killing cells based on information instead of chemistry. And we can do it relatively inexpensively. Just to put it in context, 80% of our cost of goods is DNA. And so for those of you who make virus, you can appreciate that DNA is not usually a major cost driver for virus production. But we can easily adapt to new payloads as people discover new pathways and targets. And so going forward, we're doing GLP tox, the goal of being in clinic in the first half of next year. We're doing this as a fairly large phase 1/2 trial where we're taking all comers with solid tumors. And we think we can predict what tumors will respond based on their genetics. And so in the lab, we've done this with RT-PCR, looking for the beginning of the p53 transcript. But we've also been working on a kind of a bioinformatics approach to correlate specific mutations in and around p53 with the transcriptional status. And so the goal is to take this data from the first trial, then use the diagnostic to recruit in the second. And so we can ideally call our shots in advance, and then going forward into the immunotherapeutic versions.